Hello, everyone. My name is Brianna Davis. Um, for my Journal Club 3 presentation, I will be presenting this wonderful paper published in 2023 by the Nature Journal. The title of the article is The Person-to-Person Transmission Landscape of the Gut and Oral Microbiomes, and it is written by Valles Colomer et al. I chose this article because I am really interested in how the composition of our gut and oral microbiomes is shaped. I found it really interesting that while we always talk about the other factors that influence our gut and oral microbiomes, like our lifestyle, like the things we eat, the things we do on a daily basis, I really thought it was interesting that our gut and oral microbiomes are heavily influenced by the people that we have relationships with. So we will be discussing how our interpersonal relationships can shape the composition of our gut and oral microbiomes. This paper is fascinating to me because it dives into the composition of our gut and oral microbiomes, and it poses the hypothesis that our social and interpersonal relationships play a huge role in the makeup of it. This article also suggests that microbial transmission, which is the way microbes can be shared between people, is definitely a contributing factor to how our microbiomes are made. And it suggests that since microbial transmission can contribute to the makeup of our microbiome, that it can also contribute to the study of diseases that are not caused by typical pathogens. And some people will refer to those diseases as non-communicable diseases. And so this really interests me because finding out how microbes can be shared can also lead to new development of prevention and treatment strategies of non-communicable diseases, and it can also redefine public health strategies. Why does this matter? The bottom line is that our microbiomes can play important roles in our bodies, from aiding in digestion to even how our mental health is. So, the big question, as you see on the screen, is what the authors seek to answer, and that question is, how do our interpersonal relationships and the people around us contribute to the composition of our microbiome? Using a bit more than 9,700 human metagenomes and computational strain level profiling, the authors of this article found that microbiome bacterial strain sharing across individuals have patterns, especially with mother to infant and intra-household and intra-populations. The authors of this article could not come up with a 100% conclusion on whether this transmission was direct or indirect, but it did provide a holistic overview of microbiome transmission across diverse populations, and it greatly supports the bigger question that there are many diseases right now that are said to not be contagious, and it really suggests that diseases can be re-examined with the new knowledge of microbiome strain sharing being taken into account. Let's get into the background of things. At birth, our human microbiome starts with bacteria passed down from our mothers. In an article I read by Milani et al., the establishment and interactive development of this early gut microbiota are believed or partially believed to be driven by specific compounds present in human milk. So that kind of supports the idea that our human microbiome, especially our gut microbiome, is made at birth, and it starts with bacteria that's passed down from our mothers. However, the composition of the human microbiome changes over time due to changes in our diet, our environment, our lifestyles, and especially what we will be discussing more today, microbial transmission. Since many microorganisms can't survive outside of the body for long, it must be true that the majority of these the microbial strains that we see in our gut are gotten from other individuals. In adults, the microbiome is extremely diverse, and it's completely impossible for those microbes in our gut to only be from maternal microbial transmission. It doesn't make sense for it to account for that diversity that we see in the adult gut microbiome. And this is where things get tricky, because while we know that these microbes are transmitted and spread between people and populations, it is unknown exactly how it happens and how it shapes an individual's microbiome. For this study, researchers analyzed 9,715 microbiome samples 7,646 of those samples came from stool, and 2,069 of those samples came from saliva. They analyzed stool and saliva samples from women, men, and children from several places around the world, including Argentina, Colombia, the USA, Africa, Asia, and Europe, more specifically Italy. And the purpose of them doing this was to assess the transmission across mother and infant pairs, household members, adult twin pairs, and different villages and populations. The techniques they use are pretty tricky for me to explain, but I'm going to try my best to explain them. So they performed species level profiling, which helped them to identify the species that the microbe came from, and they did that using meta pH 1A and 8, 1A and 4. And then after that, they did strain level profiling, metagenomics, using strain pH 1A and 4. If you don't know what metagenomics is, it is the study of all microbial DNA in a sample, and it really provides a way for genomes to be studied directly from their environment without having to isolate or grow them in a lab. The strain level profiling helped the researchers identify not just the species that were in the samples, but also the exact strain. Using phylogenetic distance, they were able to get an idea of how closely related the strains were, and from that, they were able to make a guess on how recent the strains have been shared with different individuals, or between individuals, I should say. They also collected metadata, which served the purpose of providing insight about the relationship between these individuals and their microbiome transmission, along with host lifestyle factors. Some of the data included age, gender, how they were delivered, so C-section versus vaginal birth, their family relationships, their location, geographically wise, and even to the point of like twin zygosity, which means they identified the strains that were shared between identical twins and fraternal twins. They also used different statistical approaches to analyze how these factors played a role in strain sharing, and how they did this was using tests like Permanova, the Wilcoxon-Rank-Sum test, the Kruskal-Wallis test, and the Spearman's test. I can go into a little detail about each test very briefly, though. The Permanova was used to compare complex data, and it provided scientists with a way to see how similar or different the human microbiome is on an individual basis. The Wilcoxon test was used when they needed to see the differences between two different groups. The Kruskal-Wallis test was used when they needed to see the differences between two or more groups. And then lastly, the Spearman test was used to examine how the strain sharing could be related to things like age or geographical locations. So now into the actual figures and the research. Figure 1, which is shown on the screen right now, represents the metagenomic framework to survey microbiome strain transmission. This figure shows the patterns of microbiome transmission and the relationship association between individuals all over the world. So in panel A, you'll see that the authors analyzed 9,715 metagenomes, and this figure really shows the data set, I'll say. So after the analysis of the stool and saliva samples, it shows that they classified the DNA found into SGBs, which I will say a lot in this presentation. And SGBs are just species-level genome bins, which help them to find the similarities in the microbial strains across different individuals. So panel A just really shows where the data is coming from, the samples they analyzed, and the relationships. Panel B shows the difference between how the microbiome can differ between humans and by their geographical location and age. The colored dots and shapes decipher between microbial communities by country and age. So you'll see that the colored dots represent the countries, and the gray circles represent people who are older than 4 years old, and the triangles represent people who are younger than 4 years old. Part C shows the phylogeny of Bifidobacterium bifidum, which is a low prevalence, highly transmitted species. And from this panel, the authors show which strains of that bacteria are found in the same person over time, which strains are found between relatives, such as mother and child, and which strains are found between unrelated people. It also goes into depth about the strains that are shared between twins and like the fathers and just other household members. At the bottom, you'll see a little graph, and it is for the purpose of showing the NGD, which I'll also say a lot, which is a normalized genetic distance plot, and it shows how closely related strains are in terms of DNA. On to figure 2, we'll talk about mother-offspring sharing of the gut microbiome. To compose this figure, the authors used 711 mother-offspring pairs across 17 data sets in 14 different countries, as you can see to the right of panel A. In panel A, the authors highlight the strain sharing rates between mother and child and the average number of SGBs in the offspring by age. The graph shows that as the child ages, fewer and fewer strains from their mothers are seen, which is really interesting because when we get to talking about the oral microbiome, you'll see that rather than decreasing over time as the child ages, for the oral microbiome, it actually increases as they age. It also shows the diversity that happens to the gut microbiome as the child grows. Part B shows SGB transmissibility from mother to infant in the first year of life. A score of greater than 0.5 means that the species is more likely to be shared between mother and infant. And using that knowledge, we can basically infer that not all strains are equally passed because some have a score closer to 0. And we can also infer that the SGBs with low scores that are closer to 0 have to be acquired from somewhere else, or they just don't have the ability to be shared. And lastly, in panel C, there are 33 SGBs that are the most highly transmitted during the infant's first year of life. So this panel divides transmissibility up into how often it is transmitted, where it is transmitted, and how long the species sticks around from childhood to adulthood. And it also gives a breakdown of westernized versus non-westernized westernized populations. As you can see, the species that seem to be transmitted more are the bacteroids and the bifidobacterium species. So the darker the blue, the higher it is, the higher chance it has to being transmitted. So this is figure 3. By putting together this figure, the authors wanted to show how living together in the same household could affect how the gut microbe strains are shared. Panel A is a Wilcoxon rank sum test, which is used to compare two different groups. It shows the strain sharing rate between individuals in the same village across 10 different data sets, which you can see to the right of the panel. From this panel, we can see that the rate of strain sharing between household members is higher than those who live in the same village, but not in the same household. Panel B shows the strain sharing rate between different relationships within a household, so mother to offspring, father to offspring, siblings, partners, and then at the end it shows the different household strain sharing rate, which is lower than the others. To my surprise, the relationships within households have this about the same strain sharing rate, and I really expected the mother and offspring to have the highest strain sharing rate just because we talked a lot about maternal microbe transmission. In panel C, it shows the relationship between twins who no longer live together and how it affects the strains that they share, or the similar strains that they shared. So this article does a good job at showing that the longer that twins have lived apart, the fewer strains they share, which makes a lot of sense because they don't live together anymore, so less contact with each other, less ability to share those microbes. The circles represent fraternal twins, and the triangles represent identical twins, and we can also see that identical twins definitely share more strains than fraternal twins. In part D, it shows the number of SGB transmissibility scores across different bacterial species. It doesn't give the exact species, but it shows that kind of similar to what we talked about the last slide. A score of 0.5 means that it's more likely to be transmitted, and a score closer to 0 means not really. In panel E, we see 21 different bacterial species. Some may look familiar from to the species that we saw on the last slide, like Bifidobacterium. These are uh, there's similarities to the last slide because these are maternally transmitted during infancy, and some of them like still stick around for adulthood. So panel E shows a comparison from those in Western populations versus non-Western westernized populations, and this is important because it really helps in the understanding of how lifestyle and environment can also play a role in like what species are shared and how they persist through adulthood. Figure 4 shows gut microbiome transmission among villages and populations. So, panel A shows a comparison of strain sharing rates between different households in a village. It also shows that even if people don't live in the same house, it's still fairly easy for these microbial strains to be transmitted. Since they're in a village, they kind of share the same lifestyle in a way, the same water, food, and environmental sources. And the dash line just shows the average rate among people in different villages of the same data set. Part B shows a comparison of strain and the strain sharing and the association to host population genetic structure. The nodes represent individuals, and the lines connecting them represent their connection by shared microbial strains or genetics. I feel like this panel is a clear indication that people from the same population tend to share the same strains, and it further indicates that transmission of the microbiota is kind of specific in a way due to environmental sources, shared habits, and relationships. Lastly, panel C shows that strains are shared within the same population, and those rates are pretty high, but not really across populations. The significance of this panel C, I feel like it shows the rarity of gut microbes being spread across populations, as I said before, because gut microbes are known to stay more within the same population. Enough of the gut microbiome. Now we'll talk about the oral microbiome and how it's transmitted between people, and we'll talk about how it is shared, if it's shared the same way or differently than the gut microbiome. But I feel like I already told you guys that it's not really shared the same way. It's kind of different in a way. So panel A compares the oral microbiome strain sharing rates across mother and offspring, father and offspring, between partners, between the same population and then in different populations. And similar to the gut microbiome, the strain sharing rates are obviously the highest between household members. But as I was telling you guys before, unlike the gut microbiome, the transmission of oral microbial strains increases with age. Panel B shows mother and offspring and father and offspring sharing rates. I'm sorry. And it shows the rates along with the median numbers of SGBs that are detected in the offspring from the ages of 0 years to 18 years. The strain sharing rates seem to increase, especially after 3 years old. And from these pie charts up here at the top, you can also see that the majority or many of the strains are shared between both the mother and father. Like one is not greater than the other, which I thought the mother would be greater. But there's also a really huge gray area, which suggests that some of these strains don't come from either the mother or the father, and therefore they have been acquired from the environment or maybe other interpersonal relationships. Panel C shows mother and offspring versus the father and offspring strain sharing rates, and they have a positive correlation. So I think that's because it's just like it's likely that if one parent has this strain, then the other does too, and that's why the child also has it because they live in the same household for I'm gonna assume up to 18 years. So this dash line across the graph, it shows that the mother offspring strain sharing rate is equal to the father offspring strain sharing rate. Sorry. And lastly, Part D shows the persistence of highly transmitted SGBs between mother and offspring between the ages of 0 years and 18 years, and also among household members who are older than 4. As you can see, over time, it just continues to increase, and I can say that I think that's because as their ages increase, they become more prone to putting things in their mouth and becoming more active and interacting with people outside their household and in different environments. This is the last figure of the article, and it shows how different microbial traits such as the gram stain, aerotolerance, motility, and spore formation can probably explain how the species is more likely to be transmitted from person to person. In the gut microbiome, gram-negative species were more likely to be transmissible than gram-positive ones, which makes sense because gram-negative bacteria are more stable. The bacteria that were motile are also more transmissible, which makes sense because the bacteria can move and colonize new hosts. But it's really unclear that, or whether or not aerotolerance and spore formation really played a role across transmission settings in the gut microbiome. In the oral microbiome, it seems as if the phenotypes didn't really matter. We only see one bolded p-value which is for the gram stain in the household box. So it suggests that gram-negative bacteria might be more transmissible as well for the oral microbiome. But these other traits, such as the aerotolerance, motility, and spore formation, didn't really have an effect on the oral microbiome. The oral microbiome seems to be more heavily influenced by social contact and environmental exposure. So I want to leave you guys with some take-home messages, take-home thoughts. So the highest rate of transmission happens between mothers and infants and people who live together. So supporting the idea that interpersonal relationships really do have an impact on our gut microbiome and our oral microbiome. Transmission can happen between people in different households who live in the same village. Again, because they're living in the same geographical location, having interactions with each other. It's easy for the microbial strains to be shared between those people, even when they don't live in the same household. They kind of live the same or similar lifestyles. Transmission very rarely occurs across different populations of people, and we can say that's due to geographical location, lifestyle differences, differences in diet, and they're not really interacting with each other because they're in different populations. Unlike the gut microbiome, the transmission of oral microbiome increases with age and are more broadly shared. We can allude that to the fact that as children grow up to be like teenagers and adults, they have more social interactions, more hugging, kissing, sharing utensils, sharing things that you put in your mouth, or just things of that nature. And from an article I read by Rothschild, published in 2018, he stated that the gut microbiome is not significantly associated with genetic ancestry, and host genetics have a minor role in determining microbiome composition. So that just covers all the bases that we've been discussing, and it really supports the idea that your interpersonal relationships really do play a role in the composition of your gut and oral microbiome. So that is the end of my presentation. I just want to say that the data presented in this article strongly supports the hypothesis that person-to-person microbial strain sharing contributes to the composition of our microbiota. I believe that the next step in this research should be tackling the mechanisms by which these strains are transmitted, and I feel like the researchers should also explore whether or not strain sharing has an impact on an individual's health, because they didn't really touch on that in the article. Overall, I do agree with the author's interpretation of the data. I feel like it was pretty straightforward in presenting the association between the strain sharing and different groups of people in their relationships. But I feel like the extra details could have helped us make a connection between association between health outcomes and disease risk and microbial strain sharing, because that is something that I would have loved to find out from this article. But thank you guys for watching. I really appreciate it. Here are 5 questions that I would like to leave you guys with and are open for discussion. How do you think these findings can help revise public health strategies in communities that are prone to diseases such as obesity that are linked to the microbiome? The oral microbiome is hugely impacted by interpersonal relationships. Do you think that targeting it could be effective for certain illnesses and/or conditions? Do you think this study helps to explain why health conditions that are not necessarily genetic appear in multiple people in a family? How might geographical factors affect person-to-person microbiome strain sharing? Knowing that our microbiome reflects our interpersonal relationships, do you think that it could play a factor in individuality? Last but not least, here are my references that I used for my presentation. Thank you guys for watching.